Good morning, everyone. It's a pleasure to welcome you to the next in our arthritis program web seminar series. We're delighted to welcome Dr. Ali Mobasheri to our seminar series, who's coming us virtually from the UK. Ali is currently the president of the OA Research Society International, or C. So welcome, Mr. President. It's great to have you with us.
Thank you.
Ali is also a professor of musculoskeletal biology at the University of Oulu in Finland, an associate professor of orthopedics and rheumatology at the University of Utrecht, and also senior research scientist in the State Research Institute in Vilnius, Lithuania, a very internationally traveled and experienced scientist. So Ali, we'll pass it over to you. It's a delight having you with us today. Welcome.
Thank you. Good morning, everyone. And it's a pleasure to be invited to join your seminar series, your webinar series, I should say. So the title of my talk is Metabolic Dysfunction in Joint and Intervertebral Disc Degeneration. And what I'd like to present to you is a story that has a lot of gaps in it. And hopefully you'll be inspired. Some of the younger scientists will be inspired to help us fill some of those knowledge gaps. So these are some of my disclosures, which also include my sources of funding, and currently I'm doing collaboration and consultancy with these companies. So it would appear that humans have evolved for 6 million years to become modern humans, and then in the current time in 2020, we appear to be, if you like, devolving into a really unique phenotype, a phenotype characterized by physical inactivity and sedentary behavior and obesity. So it's interesting as our technology gets more sophisticated, and as you can see on the cartoon on the bottom, the televisions have become slimmer, we've become fatter, and it's just a fact of life. According to the World Health Organization, obesity is a major problem across the world and has tripled since 1975. 4 years ago, when they did a survey, 1.9 billion adults 18 years and older were overweight, and of these 650 million were obese. And of course, the problem isn't just in the adult population. The number of obese children is also increasing right across the world, and the current pandemic is not helping and has not helped. It's estimated that 340 million children and adolescents aged 5 to 19 were overweight or obese, at least when the survey was done in 2016. But of course, obesity is a preventable state and can be controlled. But it's the global supply of food and our environment that's driving these changes. So this is also from the same report by the WHO showing the prevalence of obesity across the world. And you can see certain hotspots, for example, North America, interestingly, Saudi Arabia and some of the North African states really are approaching very dangerous levels of obesity, same as in Australia. But of course, these are preventable. This is a very interesting research summary and review that was published in Nature Reviews Endocrinology last year, talking about factors that can influence energy balance and cause obesity. So of course, the causes of the tipping between food intake and physical activity and factors that influence metabolism and energy expenditure are overeating. Food is everywhere. There's a buffet everywhere you go. And of course, we have lower levels of energy expenditure when we are sedentary and we are physically inactive. And there are lots of other factors tied into this story. Obesity also drives metabolic dysfunction, and it can lead to inflammation and production of pro-inflammatory cytokines by white adipose tissue. So in the scenario shown on the left-hand side, lean adipose tissue is shown with normal metabolic function. There's no inflammation, metabolic control working fine, and vascular function is working properly. And as you get increasing levels of obesity and white adipose tissue accumulation, you start to change the metabolism of the adipose tissue, producing more pro-inflammatory cytokines, including some of the interleukins, which we'll talk about in the context of cartilage and intervertebral disc degeneration, as well as some of the fat-derived, some of the adipose-derived adipokines, leptin-resisting lipocalin, etc. So this contributes to a state of elevated inflammation, decreased metabolic control, and impaired vascular function. So in summary, white adipose tissue is pro-inflammatory. And as you go from a lean state to an obese state, the metabolism of the fat changes. And as a consequence, the immune behavior of the fat tissue also changes, because some of the mediators produced in the secretome of that fat also change. So you go from having adipose tissue with a strong anti-inflammatory behavior, producing, if you like, anti-inflammatory cytokines, to a scenario where obesity is driving elevated levels of pro-inflammatory cytokines. And there are a number of cells listed at the bottom there that are participating in that process. And it's the macrophage that I will focus on in the context of both cartilage degeneration and intervertebral disc. So I don't need to read this out to you. Metabolism is what we are focusing on today. And it's basically the sum of all the chemical and biological reactions that occur within the cell. And it's within the mitochondria of the cell where all of these metabolic activities occur. And this is actually a definition provided by Hans Kornberg, who was a famous metabolic researcher. So metabolic disease is common now, and it's linked to obesity. Obesity can obviously drive increased levels of insulin resistance, type 2 diabetes, fatty liver disease, atherosclerosis, hypertension, and stroke, contributing to premature death. But it can also contribute on the right-hand side to asthma, cancer, and sleep apnea, but also osteoarthritis, intervertebral disc degeneration, neurodegeneration, and some other diseases, all also contributing to early premature death. So these metabolic diseases are mostly characterized by this high level of inflammation systemically. So in terms of obesity and what it does to the human body, in terms of relevance to osteoarthritis and intervertebral disc degeneration, we think that obesity is driving mechanical overload with impacts on the joints and on the spine, as well as elevating levels of systemic inflammatory mediators and metabolic factors that have biological activity as well. So it's not just the excess weight, it's the biological mediators that are produced by the excess white adipose tissue. There's been plenty of epidemiological work, and last week we had Tuhina Nyoji from Boston University talking about pain and osteoarthritis, but she was one of the big contributors to a lot of the studies in pain in the context of the broader story, which is epidemiological evidence for a rise in osteoarthritis, which is what Boston University is so famous for doing. So there have been various studies there looking at osteoarthritis, suggesting that obese women have a greater risk of developing knee osteoarthritis. Same for men. Obese men have a 5 times greater risk of developing knee osteoarthritis. And overweight individuals in their 30s without osteoarthritis were also at a greater risk of developing the disease later on in life. This is work by David Felsen and colleagues, suggesting that obesity can actually contribute to OA development and have impact several decades later in life. So not just increasing the risk of cardiovascular disease, diabetes, but also the risk of developing osteoarthritis in the knee. And similar impacts are also observed in the intervertebral disc and the spine. So this slide summarizes the risk factors for osteoarthritis. Obviously, age is the most important risk factor, but I've put obesity in bold there because of the relevance to this story and to what I'm going to talk about with the intervertebral disc. So age, obesity, the female gender, a previous history of joint disease and joint injuries. Obviously, there are genetic factors and endocrine factors as well. What about intervertebral disc and intervertebral disc degeneration? We know that there are similar risk factors at play here. Obviously, age is the most important one, same as osteoarthritis, but obesity makes a major contribution, as well as gender, genetics, a history of smoking, existing hypertension, and other lifestyle factors. Even diabetes is thought to be a major contributor to intervertebral disc degeneration. So there are lots of similarities between OA and intervertebral disc degeneration. I'm not going to mention back pain because I'm going to focus on intervertebral disc degeneration because the terminologies can become a little bit confusing. So what about nutrition? We know that the synovial joint, giving the knee as the example, is deriving its nutrients from blood supply in the synovium and the subchondral bone. So that's where the nutrients come from. But there are diffusion gradients for getting, for example, glucose into chondrocytes in the deeper layers of articular cartilage. And the same is happening with oxygen. So there are diffusion gradients for nutrients getting in, and obviously there's diffusions gradients for waste products and waste metabolites getting out of the cells. So the cells have to exist in a really unusual environment with low oxygen tensions, lower levels of glucose and waste products, particularly lactic acid around them. Intervertebral disc nutrition is thought to be provided by the vertebral blood vessels that provide through capillaries vascular provision to the cartilage end plate. So the cartilage end plate is a thin layer of cartilage-like tissue which provides the conduit for transfer of nutrients into the center of the disc, the nucleus pulposus. So the movement of nutrients is actually regulated by the degree of vascularity and by the biological state, if you like, the healthy state of that cartilage end plate. And similar gradients of nutrition apply here. And of course, impaired nutrition and intervertebral disc degeneration are thought to be contributing factors to the gradual loss of disc height, which you see on radiographs and on MRI, and the degeneration and degradation of the disc, especially the collapse of the nucleus pulposus. So it's thought that the cartilage end plate is a key player in providing the nutrients, and perhaps it's the loss of the nutrients to that process, which is driving the disease. So this is a very simple slide highlighting the importance of metabolism in maintaining the balance, the homeostatic balance between catabolic and anabolic activity, both in OA and intervertebral disc. Of course, if catabolic activity is outweighing anabolic activity, you run the risk of progressing disease. What we need to ensure, whether we are introducing lifestyle interventions or developing new drugs, new therapeutics, we must shift this balance between catabolic and anabolic activity and bring metabolism back into equilibrium. And metabolism can mean so many different things to so many different people working in this field. But we're going to focus mostly on the sort of classical definition when it comes to metabolism. We're going to be talking about substrates, glucose, and energy provision. So another aspect to sort of superimpose on this is the cytokine balance being perturbed in osteoarthritis and in intervertebral disc. And we know that intervertebral disc and cartilage, as their degeneration progresses, there's elevated levels of pro-inflammatory catabolic cytokines in those tissues produced by the cells within them. And there's lesser production of anabolic factors that drive matrix synthesis. And this is why some of the biotech companies and drug companies are focusing on growth factor delivery to cartilage and the disc in an effort to repair and regenerate those tissues to address this balance. This is a figure from a book chapter that I have in press with Mary Goldring and Richard Loza. It highlights the very complex scenario of the vicious cycles occurring within the joint. So a lot of the pro-inflammatory mediators that I may have called adipokines, a lot of these pro-inflammatory mediators are produced by the synovium, by chondrocytes, and also by subchondral bone. So the tissues communicate with each other, and those are important things to consider. And fragments of the extracellular matrix actually can act as signaling molecules and, if you like, active biomarkers to drive production of additional cytokines and chemokines and really perpetuate this vicious cycle. So in terms of adipokines, the pro-inflammatory cytokines that come from white adipose tissue in the obese state, we now know that these cytokines contribute to metabolic syndrome. They impair cell function. Obviously, there's excess weight in obese individuals, and that is contributing to the biomechanical changes in the tissues. So here I've only highlighted the synovial joint, but the same applies to the intervertebral disc. Excess weight will have similar impacts there. And the adipokines will contribute to a state of chronic low-grade inflammation, altering the biological activity of the cells and tissues within the joint and the disc. So mitochondria are key components of this metabolic story. Mitochondria are what produces cells, energy within the cells in the form of ATP. Obviously, energy is initially derived through glycolytic breakdown of glucose, but then that's not very efficient. It produces much less ATP compared to oxidative phosphorylation, which happens within the mitochondria through this oxidative phosphorylation system indicated on this slide. And this can generate 36 ATPs from a single glucose molecule. Now, we know that mitochondrial dysfunction is a key feature of rheumatic diseases, not just osteoarthritis, but many other forms of rheumatic disease, inflammatory arthritis, systemic sclerosis, fibromyalgia, gout, etc. So impaired mitochondrial function, and if you feel like mitochondrial dysfunction, is driving a lot of these diseases because of some of the underlying mechanistic changes, oxidative stress, reduced levels of mitochondrial biogenesis, involvement of mitochondria in cell death, nitric oxide production, impairment of the antioxidant system, and obviously there's some contributions from the mitochondrial haplotypes as well. But of course, the story of metabolism starts with the substrate, and the substrate is glucose, and glucose has to be carried into the cell. It doesn't diffuse across the plasma membrane. There are glucose carriers in the plasma membrane, the GLUT transporters that bring it in. And in the pancreatic beta cell, we know that when blood glucose rises, that's a very important metabolic signal that closes KATP channels, causing membrane depolarization, activation of voltage-gated calcium channels, free calcium entry, and insulin release from the pancreatic beta cells by the process of exocytosis. Now, we know that the University of Toronto has been a pioneer in this area of metabolic research, and the work that was done at the Banting and Best Institute in the University of Toronto led to the discovery of insulin as a key hormone involved in glucose regulation, and eventually the production of the very first biological drug, which was human insulin. In 1982, recombinant human insulin was the very first biological drug that was introduced. So it had a very interesting story, which is highlighted in this slide. I won't tell you all of it. I won't read the slide. I'll simply say that Frederick Banting was an orthopedic surgeon, and he had a massive impact on our understanding of metabolism. And Banting, Best, and their colleagues, the broader set of collaborators, without the help of collaboration of the biomedical scientists and the veterinarians, they would never have got this work done. And obviously, the rest of it is history. So in terms of glucose sensing, the pancreatic beta cell is absolutely critical. And then in the gut, there are various mechanisms for sodium-dependent uptake of glucose from the lumen of the gut, and then sodium and glucose come in, and the glucose is either used for metabolism or is shunted across the cell and ends up in the circulation through GLUT2 glucose transporters. So we've shown over many years that chondrocytes have a number of glucose transporters that are involved in the initial step of metabolism, the uptake of glucose. GLUT1 and GLUT3 seem to be particularly important. And of course, glucose is an important energy and supply for chondrocytes and a precursor for the production of glycosaminoglycans. So the work started when I was a young lecturer, assistant professor level at the University of Liverpool, and I was fortunate to work with a group of scientists working in the connective tissue research group there, led by Stuart Carter, and Steve Richardson was a promising young PhD student who helped with a lot of these experiments. And with his help, we were the first to show that chondrocytes express GLUT1, GLUT3, and GLUT9, at least healthy, normal chondrocytes. And these glucose transporters, we provided evidence for their molecular expression. This is long before quantitative PCR. We were using conventional PCR there, immunofluorescence and FACS analysis. And we provided the first evidence that GLUT1 and GLUT3 are major glucose transporters, but GLUT9 is also present in these cells. And Steve Richardson continued this work, and we were able to identify more of the isoforms of the glucose transporter in chondrocytes. And we were able to show that it's regulated, the system's regulated by growth factors and by cytokines, by insulin, insulin-like growth factor, and pro-inflammatory cytokines as well. We found evidence for multiple GLUT isoforms in chondrocytes, and we started doing immunocytochemistry to show changes and alterations in disease. But importantly, we also started doing some functional experiments looking at the uptake of tritiated glucose, 2-deoxy-D-glucose, and we showed temperature dependence of it, as shown here in a figure on the right-hand side, and also the fact that uptake of tritiated glucose is stimulated by insulin, by insulin-like growth factor, recombinant form of that, and cytokines. So it seemed that cytokines and growth factors both drive increased uptake of glucose in chondrocytes. And of course, the system is the same that you would find in other mammalian cells because it's inhibited by the glucose transport inhibitors, cytochalasin B and fluoretin. When I moved from Liverpool to University of Nottingham, I was becoming more and more interested in developing proteomics and particularly membrane proteomics methods. And we started looking at the membranome, the surfaceome of isolated chondrocytes. And we developed methods to fractionate proteins from chondrocytes. And in some of the crude early experiments, we were using detergent-based hydrophobic versus hydrophilic separation systems to identify some of the membrane proteins. And even this crude approach resulted in identification of GLUT1, GLUT3, and another GLUT isoform, GLUT14, as fairly abundant proteins in the plasma membrane of chondrocytes, along with some other membrane proteins that hadn't been identified before. And of course, we provided immunocytochemical evidence for this in the isolated cells as well. These are not cell lines, these are freshly isolated. And the work continued in the hands of a brilliant Marie Curie fellow from Hungary, Dr. Csaba Mata, who's now gone back to his native Hungary, but in our lab, he developed a surface enrichment method for labeling the surfaceome, and we continued this work, and we've just published another paper looking at alterations in the chondrocyte surfaceome in response to pro-inflammatory cytokines. I won't go into the details of this, but we found extensive changes in the surfaceome, in the membranome of the cells following cytokine stimulation. So you can imagine a scenario where presence of cytokines and adipokines can actually really drive phenotypically important changes in the chondrocyte. So this slide summarizes some of our observations. Not all of them have been published. When we do quantitative proteomics, we see that there are changes in the membranome in response to those pro-inflammatory cytokines at physiologically relevant concentrations. Glucose transporters are upregulated, and many others have made these observations, particularly the group of Martin Lotz and Armin Schickman at the Scripps Research Institute in La Jolla. Sodium potassium ATPase, another important metabolic switch, is downregulated. Integrins and catecherins are downregulated, and there are changes in some of the other proteins. I won't read all of this slide to you, but particularly there are changes in the expression of vascular adhesion molecules, and these are upregulated in the presence of cytokines. We went on further to show that potassium ATP channels, KATP channels, are important sensors of intracellular ATP levels in chondrocytes, and they can respond to changes in extracellular glucose as well. And that was work done in collaboration with colleagues at the University of Coimbra in Portugal. And we were also able to show that cytokines are also regulating not just the glucose transporters, but the KATP channel. So the KATP channel is an important component of the glucose sensing apparatus in chondrocytes. So I'm going to move to immunometabolism and talk about immunometabolic changes in disc and joint disease. It is now clear that cellular metabolism is important for regulating immune cell function, and immunometabolism is where you have metabolism meeting immunology. So we know that there are immunometabolic alterations in osteoarthritis. We've discussed this before: lifestyle changes, obesity, poor diets, existing of comorbidities such as type 2 diabetes, hypertension. They can all contribute to metabolic reprogramming, not just in the liver and other vital organs, but also in the joints and in the skeletal system, and contribute to inflammaging and early senescence, what we term as chondrosenescence, leading to osteoarthritis. So under normal conditions, in a healthy cell, you have a good balance between ATP production via glycolysis and oxidative phosphorylation. But when you have immune cells activated, and it's particularly focusing on T cells, this research, there's elevation of glycolysis, which is less efficient, but it happens to be increased compared to oxidative phosphorylation. So immune cells, when they're activated, they prefer glycolytic energy provision. And there are important bioenergetic differences between M1 and M2 macrophages. M1 macrophages are pro-inflammatory, whereas M2 macrophages are anti-inflammatory. M1 macrophages are glycolytic, but M2 macrophages are oxidative. So their biological behaviors are different as a consequence of their metabolic behaviors. And there's evidence that there are changes in the TCA cycle and M1 macrophages. Particularly, there are breaks after citrate and succinate, resulting in accumulation of citrate and succinate, which actually have biological consequences, regulating HIF1 alpha, accumulating HIF1 alpha, inducing IL-1 beta. That's in the case of succinate, and in the case of citrate, driving changes in fatty acid synthesis and in nitric oxide production. So these metabolic breaks leading to accumulation of those metabolites, they're not just metabolic events that you can deal with. These metabolites actually have biological consequences, and they have downstream impacts. So they are using different pathways. The inflammatory M1 macrophage is using more glycolysis, and the M2 macrophage is using more of the TCA cycle. So that's a critical difference. Then there are some of the features of effector T cells, regulatory T cells are also highlighted here in this slide, but I'm not going to go into that. What about chondrocytes? We know that chondrocytes are glycolytic cells because of the avascular nature of articular cartilage. The same is true in intervertebral disc. They rely more on glycolysis anyway, but it turns out that in altered states such as osteoarthritis, in disease, in the presence of cytokines, the glycolysis is even more perpetuated. So there's less emphasis on the TCA cycle, and there's evidence that mitochondrial dysfunction actually has some negative impacts on the regulatory pathways that sustain energy homeostasis. So metabolism in chondrocytes in osteoarthritis is altered, and that's driven by pro-inflammatory cytokines. So we thought, can we model this? We used the seahorse analyzer from Agilent. It's basically a sophisticated oxygen and pH electrode. So basically what it does allows you to measure oxygen consumption rate and lactate production, production of protons, and plot those with time and look at cells treated with different biological reagents or drugs. So the instrument produces these typical curves, allowing you to measure basal respiration. And then you can use some pharmacological mediators to inhibit various processes and calculate ATP-linked or proton-linked respiration. FCCP is an uncoupler that allows you to measure maximal respiratory capacity. It's a really important measure of the cell's ability to cope with stressful situations. So if the cell's really robust at dealing with a stressful environment, it has a high maximal respiratory rate. So the instrument is really useful in allowing you to measure all of these things. So we've done some experiments with the seahorse to show you that actually we can get typical responses as you would get with any other cells with primary chondrocytes. But the interesting thing is that the moment you add cytokines, you completely blunt those responses. So chondrocytes pre-stimulated with cytokines, whether they are stimulated for short periods of time or longer periods of time, they actually have lose their ability to generate this spare respiratory capacity. And it seems that interleukin-1 beta is a key driver of that. So we've done experiments where we've used IL-1 by itself and IL-1 with other, in combination with other cytokines. You can clearly see here the interleukin-1 beta is the driver of that blunting process. So interleukin-1 beta is like taking a sledgehammer to the metabolic machinery of the cells. So it has really profound impacts on the ability of chondrocytes to generate energy and respond to that stressful catabolic inflammatory environment. We don't know much about the disc, and we're hoping to study the biology of the cartilage end plate in collaboration with my colleagues Osmo Tervonen and Jaro Karpinen at the University of Oulu. My colleagues are experts in modic changes, measuring modic changes in the intervertebral disc. And in collaboration with them, we're going to be looking at the cartilage end plate through a variety of techniques, including micro-CT, Raman spectroscopy, proteomics, and metabolic profiling, because these experiments need to be done with end plate cells. They have been done with the intervertebral disc cells, and there are subtle similarities between them and chondrocytes, but I don't really have enough time to go into more detail. And also in collaboration with Oreste Gualillo at the University of Santiago in Spain, we are interested in looking at the other adipokines that influence the different cells of the intervertebral disc. So this is all work that's planned for the future. So I think I'm reasonably close to time. I maybe just 5 minutes over. In summary, metabolism has a key role in the physiological turnover of articular cartilage and intervertebral disc. In osteoarthritis, chondrocytes and other joint cells undergo metabolic alterations in the presence of the cytokines and shift from a resting regulatory state to a highly metabolically active state, but it's not very efficient because it's more relying on glycolysis. The levels of inflammatory mediators go up, the metabolic intermediates accumulate, and the cells of the immune system within the joint can also be activated in that scenario. And we believe these are understudied areas of joint biology, and I think we need to focus on them more, because some of the key metabolic pathways could eventually reveal some interesting targets for intervention. And of course, there's no better intervention than diet and exercise, but of course, we need to learn more of the basic science of the metabolism of these tissues. Of course, I'd love to be there in person to give this seminar face-to-face. I had the pleasure of being invited to University of Toronto for examining Ghazal Tavalai's PhD back in January, and I had very fond memories of my trip. And Mohit, I've left a cup, a mug, and some sweeties in your office. I hope you haven't eaten them all. When I return to Toronto, I'm going to be claiming them. Thank you very much for your attention.
Thank you, Ali. This was a terrific overview. By the way, I've already eaten those sweets which you left here. So.
Oh, naughty Mohit.
Just a reminder to the audience, please post your questions on the question and answer tab on Zoom. First question is by Dr. Jason Rockel. Cyclic AMP is an important molecule that can modify cellular metabolism, in particular, increasing lactic acid release. There are many GPCRs that can modulate cyclic AMP, particularly in response to a variety of ligands. Were any GPCRs modified at the cell surface by inflammation?
That's a great question, Jason. We haven't focused on GPCRs, but we have a data set. We have quite extensive proteomic data sets. We can start to investigate those. We actually started focusing, because we're interested in metabolic pacemaking and potassium channels, and we're also interested in some channels that are involved in chondrocyte apoptosis. We focused on those first, and trying to make a story out of that, but of course data is available, we can share with you. Also, in your comments, you said lactic acid acidifies the local chondrocyte environment, potentially modifying protein function. How does that contribute to OA? Well, we know for decades that levels of elevation of lactate within both cartilage and intervertebral disc actually result in reduced proteoglycan synthesis. I think some of those experiments were done many years ago by Alice Marudis and Jill Urban. So lactate really has the capacity to drive down metabolism and matrix synthesis. So that's the second part, response to the second part of your question.
Thank you. A question from Amanda Alley. Thank you for an amazing presentation. My question pertains to the role of various adipokines. You mentioned leptin and resistin as pro-inflammatory factors in OA. What are your thoughts about adiponectin? Is this a good guy or a bad guy in OA?
That's a really good question, Amanda. Thank you. I think, to be perfectly honest with you, all of the molecules that we're talking about, we can't just say they're good guys or bad guys. I think they have context-dependent roles. So we all know that interleukin-1, interleukin-6, TNF-alpha, they perform really important biological functions, and without them, you know, immune regulation would not be possible. But of course, we know that in some scenarios when we're studying these things, we see elevation and increased levels, and we label them the bad guys. So I don't think that adiponectin is always a bad hombre. It's possible that some of the literature portrays it in that way, but I think its role is context dependent. I hope that answers your question.
Thank you.
Ollie, talking about the metabolic state, in a joint we have distinct tissues, and of course they will probably have distinct metabolic state as well. How does
Sure.
that actually contribute towards the overall pathology, whether it's related to disc degeneration or it's related to, say, joint degeneration, in terms of osteoarthritis or any other?
Thank you, Mohit. I think it's important to consider that the metabolism of synovial lining cells, fibroblasts, is very different to metabolism of chondrocytes. They are very different cell phenotypes. And the same for subchondral bone cells. So different tissues have different metabolic states and requirements, nutrient and nutritional requirements. And I think that a lot of the gaps remain. We haven't done a side-by-side comparison. We certainly haven't done side-by-side comparison of metabolism of chondrocytes versus primary synoviocytes versus macrophages from the joint. So I suspect, Mohit, there will be different. And I suspect that in a normal, healthy, functioning joint, there's a lot of buffering happening. So the lactate that I discussed in the previous question, there's probably a much greater capacity to deal with the buffering of lactate and some of the other factors that can potentially impact on matrix synthesis. But we haven't done these studies properly side by side.
Question from Somio Viswanathan here in Toronto. Ali. Hi, Ali. Great talk as usual. Very interested in the studies that showed that inflammatory cytokines, particularly IL-1, affects mitochondrial function, oxygen consumption, and spare respiratory capacity. Why does targeting IL-1 then not reverse this? Is it too far upstream? Do you think that reprogramming of mitochondrial function at the cellular level, say at the chondrocyte or synoviocyte, is necessary to reverse this metabolic dysfunction?
That's a great question. Yes, I mean, I think that obviously targeting, I suppose in your question, when you mentioned targeting IL-1 beta, you probably mean targeting it in a clinical trial with neutralizing antibodies. Is that what you mean? I suppose that's what you mean. That's what you mean. I'm not really sure that those studies have the capacity. Those approaches have the capacity to completely neutralize IL-1 within the joint. I think this is quite an interesting story. If you think about the anti-IL-1 therapies that have been applied, my understanding is that in the most recent clinical trial, interleukin-1 beta was injected subdermally. So the hypothesis is that we are taking a biological drug, a biological approach to a systemic inflammatory disease and applying it for OA. But I don't think they injected the anti-IL-1 beta and neutralizing antibodies into the joint. I think they put it under the skin. So you're actually relying on diffusion of the antibodies out of the skin, getting into circulation, and then penetrating the joint. I'm not really sure if that's the best way to design a trial focusing on osteoarthritis in one or two joints. I'm not really sure that's the best way to go. And the second part of your question, do you think that reprogramming of mitochondrial function at the cellular level, at the chondrocyte or even the synoviocyte is necessary to reverse this metabolic dysfunction? I think it is possible to reprogram mitochondria. We know from lots of studies of skeletal muscle, training skeletal muscle, or increased physical activity, you can reprogram metabolism and mitochondria by changing behavior and changing physical activity levels. So it is possible to do. I'm guessing it might have an impact on the joint as well if you take in those systemic interventions through physical activity and behavior. I wonder if that answers your question, but of course, ask me if I haven't.
Next question is by Dr. Arthur Bookman. Is there a direct interface between exercise, weight bearing, and glucose uptake by cartilage? And how does this occur?
That's a great question. I haven't seen the papers. I haven't seen the evidence for that. I would imagine that increased levels of physical activity, of course, result in elevated levels of glucose uptake by muscle. And muscle has GLUT1 and GLUT4 glucose transporters. GLUT4 is actually insulin responsive glucose transporters, and physical activity trains your muscle to buffer glucose much better. So that's why physical activity is so beneficial for muscle function, but I'm not sure those experiments have been done in cartilage. In fact, I'm sure they haven't been done. But if you have seen the papers, please let me know.
Ali, I had a question. There's quite a revolution, as you know, in the treatment of type 2 diabetes. We've got GLP-1 receptor antagonists now, sodium glucose transport, SGLT2 inhibitors like Jardiance. And I'm wondering whether any of those novel therapeutic agents in type 2 diabetes have been applied in the metabolism profile of the IV, either the intervertebral disc or the osteoarthritic joint.
Dr. Inman, that's a great question. I don't believe they have, but I think they should be. SGLT, I don't believe SGLT is expressed in chondrocytes. Or if it is, it hasn't been extensively studied. I would hypothesize that SGLT should be expressed in chondrocytes because it should be expressed in any cell that has the capacity to use a steep inward sodium gradient. So you know that the extracellular matrix of both cartilage and intervertebral disc is so rich in proteoglycans and glycosaminoglycans, and these are counterbalanced by cations. So sodium is very concentrated in cartilage and the disc, and the inward gradient into the cell is quite steep, quite significant. So I suspect that SGLT, multiple SGLTs may be expressed in chondrocytes. I haven't looked at the literature specifically for SGLTs, but I would imagine that that's probably worth trying. I think synoviocytes do express them, but SGLT for many years was thought to be just exclusively expressed in the lining of the gut in enterocytes. But I think it's a very, very good target for regulating glucose balance and obesity.
Thank you.
Ollie, if we were going to initiate studies understanding metabolic dysregulation in biospecimens from patients, for all our young scientists, can you actually give us any insights on how we design our new experiments to be able to really understand metabolic dysregulation, especially when we're looking for biomarkers, looking for therapeutic targets?
Okay, that's a great question, Mohit. I think I would always start with cell models and tissue models and work up to preclinical models, as your lab and many others have done. I think my advice to the young investigators is set up a really reliable and robust in vitro model, test your hypotheses in that model, and then move up to preclinical models before going to cohorts. And if you are designing human cohort studies, phenotype them really well in terms of their diet and physical activity levels and whether they have other comorbidities. I know that all the ongoing studies are now focusing on trying to understand, using machine learning, what are the links between existing metabolic disease and the progression of osteoarthritis. I haven't mentioned all the really nice work done by Francis Berenbaum and colleagues implicating diabetes and metabolic changes, metabolic set points in osteoarthritis. I think those are really important contributors to a cohort study in humans, especially if you want to study their biomarkers, systemic biomarkers, and microRNAs, etc. But I think the advice to the young scientist is always address your hypothesis in a really robust basic system, and then expand out to more complicated, broader populations.
We have a question from Angela Chung, Ellie. Thanks for a great talk. Have you used PET FDG imaging to study joints as it relates to metabolic activity?
That's a great question, Angela. No, we haven't. That would be really great to do. Of course, you know, these kind of PET studies are mostly focusing on uptake of glucose by tumor cells, and there's a whole body of literature on how you can use PET FDG to image tumors. And I believe there may have been 1 or 2 studies on chondrosarcomas and osteosarcomas, but we've never studied that healthy joints or cartilage using this approach and linking that to metabolic activity. I think it probably needs to be done, and it's probably all in the future. I think, I'm sure, I'd be surprised if somebody's not doing it already, because it seems like the obvious thing to do. Oh, and Angela says we've used it to study bone formation and inflammation. Yes, so we just have to extend that study to cartilage and to the synovium to see what's happening there. I think those are important experiments that need to be done. And I don't know how the metabolic activities in cartilage and bone will compare. Of course, bone metabolic activity will be higher because it's better supplied with blood vessels. So there will be metabolic differences between cartilage and bone. Actually, Angela, I'd like to tell you a very, very brief story about how I started life looking at chondrocytes and membrane transporters and glucose and metabolism, etc. I went to a conference in the US. It was a physiology conference, and a very eminent professor of physiology came up to my poster, and he's looking at it saying, chondrocytes. He was a very eminent professor at Yale University. He said, oh yeah, I know about these cells. But these cells are dead. They shouldn't have any metabolism. How dare they express glucose transporters? So from a certain point of view, he was right compared to kidney, compared to cardiomyocytes, to proximal tubule cells, expression of glucose and metabolic rates of chondrocytes are much lower. And we know that with development and with aging, that diminishes even further. So metabolism is quite slow in chondrocytes, especially compared to other tissues. But I think that in itself is a hint that we should be studying it, because we know that it gets impaired with aging, and there's a possibility that we could find ways to modulate metabolism for anabolic benefits to ensure we have better regenerative strategies for cartilage and other joint tissues. And I think that's really the important link. Linking metabolism to anabolic activity and to cartilage regeneration. And perhaps some of the future D modes, they will be not just having an impact on pain and an impact on structure and tissue regeneration. Maybe there will be an impact on metabolism and anabolic growth and regeneration of cartilage, as many of the growth factor-based approaches promise to do.
I had a question, Ali. You showed quite profound changes in the chondrocyte biology as a result of pro-inflammatory cytokines. The intervertebral disc, as it is relatively acellular, avascular at any rate, the question really ahead was really what likely is the source of those cytokines? Is it thought to be likely the subchondral bone marrow? And if so, is that really where the problem begins, in the subchondral bone marrow?
I think the cytokines, there is evidence that nucleus pulposus cells have the capacity to produce, at least at mRNA level, cytokines. But I think you're right. The cytokines are probably coming from bone marrow. And if you remember at the beginning of my talk, I was hypothesizing that in a situation where you have obesity and elevated levels of systemic adipokines, I would imagine that those would be higher and perhaps penetrating at higher levels into the disc. So I think the hypothesis is that one of the most interesting hypotheses right now in disc pain and disc biology is about bacteria and toll-like receptors. There's the idea that bacteria are driving a lot of the changes, and bacteria, particularly anaerobic bacteria, are linked to low back pain. But also the idea that elevated levels of pro-inflammatory cytokines in sedentary individuals who don't have much physical activity are also driving metabolic and phenotypic changes in the disc, not just in the nucleus pulposus, but also perhaps on the cartilage end plate. Maybe the cytokines systemically are actually impairing the metabolic set points of the end plate cells, and therefore impairing with the diffusion of nutrients into the center of the disc. That's the hypothesis anyway.
Thank you.
Looked into mitochondrial respiratory states in the chondrocytes.
Mohit, could you repeat that, please, one more time?
Have you looked at mitochondrial respiratory states in the chondrocytes?
Yeah, you know, with the seahorse instrument, we've actually looked at mitochondrial function as well. There are various off-the-shelf kits that you can buy from Agilent to look at mitochondrial respiratory capacity. So there's the MitoStress kit. So yes, the chondrocytes exposed to multiple pro-inflammatory cytokines appear to have impaired mitochondrial activity. So these cytokines have an impact on mitochondrial function for sure.
Wonderful. I think we have no more questions, Ali. Thank you so much for a wonderful overview. Just a reminder to the audience, please join us next week as well for our next lecture, which will be lecture number 10 by Dr. Mary Crow. And it's going to be on interferon and lupus as a mechanism and a target. This is going to be on July 28th at 8 a.m. Again, thank you so much, Ali, for joining us and taking our time, and see you all next week.
It's a pleasure. Thank you.